Welcome. I'm Julie Dragon from the University of Vermont, and on behalf of myself and my colleague, Emily Curd-Guswa, I'd like to share with you how we are using ONT to shine a light on a dark mystery, that of the occurrence of melanoma in benthic or bottom-dwelling fish in Lake Memphremagog. First, I'll give you some background on the problem, and second, I'll describe the data we have so far gathered to address this problem. So the problem is that melanoma is found consistently in 30% of one species of fish in one lake in Vermont. This species is a type of catfish called the brown bullhead that lives in and among the plants and mud at the bottom of a deep lake called Lake Memphremagog. This is a higher than usual rate for melanoma in any species of fish, even those that live in shallow waters in sunny tropical locations, which Vermont is definitely not. Lake Memphremagog is a transcontinental lake shared by Vermont in the United States and Quebec in Canada. The lake is popular for recreation and fishing and is an important source of water. It's 31 miles long, with 73% of its surface area in Quebec, where it provides potable water for 200,000 people. However, most of the watershed that feeds the lake is located in Vermont and is the source of accumulated phosphorus, sediments, and other pollutants. Brown bullhead have been found with various types of melanistic lesions, as you can see in these images on the right, from simple punctate dots in A to multiple raised tumors, like in D through F. Fish of various kinds are known to get melanoma. However, most often it's found where fish are exposed to high levels of UV light, as mentioned, in the shallow waters or surface-dwelling fish and in sunny locations. In this lake, these lesions are never seen in young fish, nor in other fish species in the lake. Within the species of fish, lesions are infrequently seen in other lakes within its range, which includes the Great Lakes, east to the Atlantic Ocean, and all of the Mississippi Basin. Lesions may be associated with maturation in fish, though never appear to be lethal, and increased reports of these lesions initially came from anglers finding these fish coincident with a major flooding event in the region in 2012. Various theories have been put forward about the issue and focus mainly on contamination-induced disease from either chemical or microbial pollution. Public interest is putting pressure on local government to address the problem. It is known that a local landfill has been leaking leachate into the lake for some time, and public interest groups blame this landfill for putting forever chemicals, PFOAs, and the like into the lake, but without much evidence for a higher than average presence of those chemicals in the lake or in the fish. Local government scientists came to us to help them find some answers. Our colleagues hypothesized the cause to possibly be chemical exposure, or virally related, or due to particular genomic background of fish that spawn in the same location year after year, or some combination of the above. However, without a reference genome, questions of genomic background or viral integration could not be addressed, and previously collected transcriptomic data were not very informative. We're just beginning to gather data from about 200 fish collected so far, with more coming this fishing season. We started with building a reference genome, since there was none, with brain tissue from 8 bullhead from lakes and rivers other than Lake Memphremagog. And we then sequenced genomes of tumors and normal brain tissue from 10 affected fish in Lake Memphremagog, plus 10 unaffected fish out of 40 planned for whole genome sequencing, also from Lake Memphremagog. And from these data, we will address population genomics, germline and somatic variation, and differential methylation. And we've used the newly assembled genome to analyze the older transcriptome data collected back in 2016. We don't currently plan to take a deep dive into microbiome or viral integration, though our colleagues would like us to, as their original hypothesis was that a particular RNA virus was the cause of this affliction. We've used a combination of long and short read sequence data thus far, with the long read data coming from the Promethean 2 solo, and we've assembled the genome with Minnow and a custom pipeline and annotated it with Fun Annotate. And we've analyzed for methylation status using DeepMod2. Coverage and quality was not even over all the reference fish, but generally good. And one fish was particularly good, which is highlighted in red, with 73x short read coverage and 136x long read coverage, and strong metrics supporting the quality of the assembly and the annotation. The closest reference genome had been the channel catfish, but our assembled genome is a bit larger at 859 megs and contains 30 chromosomes, which is one more than the channel catfish. We worked with an enthusiastic undergraduate student to build a custom pipeline to perform all the tasks needed for this non-model assembly, and he named it the Pegasus pipeline. It incorporated the channel catfish genome to assist in scaffolding the bullhead genome, as well as the previous bullhead transcriptomic data and a benchmark fish gene set to assist in predicting coding regions in the annotation step. Using our assembled genome and whole genome sequence variant analysis from Lake Memphremagog fish, we calculated a minimal spanning network that clearly distinguishes the reference fish in the purple circle from the Lake Memphremagog fish. And within Lake Memphremagog fish, the unaffected fish and affected normal tissue in the gray circle from most affected tumor tissues in the black circle. We'll be adding more reference fish and unaffected fish to this analysis this summer. Based on posterior probability of membership in a location due to shared variants, each sample's similarity to another is described in this chart. The reference fish have a purple border, and the analysis indicates homogeneity in two reference fish populations and mixture between the other two. Lake Memphremagog tumor normal pairs, which are bordered in black, are more like each other than other tumor normal pairs from the same location. And Lake Memphremagog locations for which unaffected fish were included, which are starred, are often more like affected fish in their location than each other. And lastly, some evidence of mixing of locations is seen. Comparative gene expression clearly distinguishes unaffected fish from affected fish, even within Lake Memphremagog. In this PCA on the left, fish from another lake are in orange, Lake Memphremagog unaffected fish in green, and affected fish in blue, and showing a strong transcriptomic signal from the affected melanistic fish. Many upregulated genes in tumors are associated with melanocyte differentiation, DNA repair, and tumorigenesis. We did look for the suspected RNA virus in these data, but didn't find it. Looking at somatic variants, we see missense SNPs are enriched in affected fish, not surprisingly, and these are most often C to T transitions. And 20 genes with the most variants are implicated in tumorigenesis and immune function. Mutational signature analysis of these variants showed an enrichment in the signature SB40, which is not associated with chemical or UV exposure, as we thought it might be, but does have an association with cancer generally. We've only just begun to look at differential methylation in a subset of our affected fish, but we do see increased methylation in affected tissues. We have found one shared region of increased methylation across all 5 examined fish so far that involves a hypothetical protein with G protein coupled receptor activity. GPCRs are transmembrane proteins that transduce external stimuli into cellular response and are known to be involved in controlling initiation and progression of tumors. A methylation of these sites has been found to be widespread in pan-cancer studies. This is work as a result of a tremendous team effort, and I'd like to acknowledge all of my colleagues for their help. And thank you for your attention.